Sam is a little concerned because he thinks that I'm just going to stand up here and start rambling and making him uncomfortable. So I'm going to do just that. So welcome to the defense of the great Sam Gould. So he officially joined the lab in May 2022, and then we're in December 2025, and he's unfortunately, but also very excitingly, defending his thesis. I want to give you a bit of a different story. You can read all the facts about him and talk to him afterwards. But actually, I first met Sam back in New York, in Manhattan, in New York City, back at Sloan Kettering, where he was a summer student in the SERP program. And as you can see him back there, it's a very happy, a very high resolution picture of him. And he actually presented a very nice poster at the end of that summer research experience. By the way, the reason that I know him from back at Sloan Kettering is that he was a summer student in the lab that I would train as a postdoc. He didn't work with me. He worked with a good friend of mine called Kaloyan, Kal Sanov. And here they are presenting this very nice poster. He's always been very funny at only a few, random times. But one thing that I think people in the lab are surprised to see is that there were actually some Western blots in this poster. And I think if you look at the actual poster, you can get an idea of where his choices for different colors come and how he's evolved to use the right reds and the right blues throughout the years in the lab. And here he is getting his formal diploma at the end of the summer research experience. And I just want you to know that Lydia Finley, who was a PI at Sloan Kettering, was definitely much taller than little Sam here, which then turns into this potentially angry Sam, or baby Gould, as he would then later become an emoji immortalized in our Slack channels back in May 2023. Okay, so now getting into the more serious stuff. So I got this email from Sam January 10th, 2022. It's like, hey, I'm not a doctor or anything, right? So just, hey, Francisco. But this is really one of the luckiest days, of my life and career, and I think everyone in the lab. And you don't need to read the whole thing. It's like, hey, I heard you're at MIT, and I'm interested in talking more about research. Notice that he was even using an exclamation mark, which is very unusual. And he was already using dashes for some reason, probably using ChatGPT to write it. And then he sends this long email. If you know Sam, he's not the type of person that sends you a long email. So I was surprised by this, and it's probably still the longest email I received from him. There were no exclamation marks. He proposes a project, and projects for the future. And then things become official. He becomes a rotation student in January 2022. And this is us. Lab was empty, had just opened. Still dusty. Alex, who was the other person that initially joined the lab, actually originally in January was not even there. And again, you're like, Sam wearing a mask? It's not a thing. But then fast forward, he ends up joining the lab, working on papers when he's not officially even a member of the lab yet, and then proceeds to put together what then later became our lab's first preprint. And this was only 4 months into his PhD, which is very impressive. And then he was just getting started, and he has I was trying to count more than 10 publications that are in press, in review, or published so far. He has represented the lab many times, very proudly. This was our first presentation of the Colerain meeting. We later celebrated over mimosas, wearing more like business casual or formal. And I was surprised why Sam would want us to wear these clothes for a brunch right over there, actually, in Cafe Luna, which is a minute walking. But then I saw this in his desk. Turns out that all this time he was also a contractor at Pfizer. So he was having a side gig. And so that's when I started to understand this Sam of the work-life balance and this very successful human being. But this is the actual picture in Cafe Luna with Grace and Sam and Angel, who were the initial members of our lab. And these are just some examples of some of the adventures over time. Now, again, I know I'm rambling, but this is worth it. I want to also emphasize something that has always impressed me from Sam. This is me sending him a message when I'm at a Cold Spring Harbor meeting at 8:10 p.m. or something, maybe it was a Saturday. And I said, I talked to this editor, and I said, let's get ready. And he's like, let's go, censored. And then at another time, I think this was when working in his prelim proposal, I said, yeah, maybe I'm going through his comments. He says, maybe I'll do this tomorrow. He said, unless there's a deadline. And then I said, yeah, there's a deadline. It's tonight. And he's like, all right. And then this is actually became this picture slash video that Sam sends me when we're on a time crunch, which is the exciting time. And then there's like a series of text messages that actually cover different areas of our relationship over the years. One is that he forgot to put a stop codon on a cDNA. The other one is that he apparently was thinking about the gains while he was giving a presentation. I get pictures from Django, his dog, every now and then, and I feel like he's my dog in a way as well. And then we talk about art. He loves Rothko. And then we also have this, interactions that are very nice related to UFC and betting, and so we're texting at weird times of the night sometimes to see like how we did on certain bets. But again, he's been just a pillar of the lab, very proud. I am very proud of him, and I think all of us are super proud. And so, thank you for making the lab a reality, and really looking forward to the day that I was dreading, and I even wrote this in the letters of recommendation, the day you defend. So, Sam.
Thank you, Francisco, for the very kind introduction. I'm not gonna lie, I think I was the most worried about that part. I wasn't sure what he was gonna say. It was mostly nice though, so that was nice. I'm gonna hold my acknowledgements until the end of this talk, but I do just wanna start by saying that MIT has really been an amazing place to do my PhD, and I wanna thank Francisco, all my coworkers and lab mates, my thesis committee, and all the faculty and staff and scientists here that really make it such a special place to do research. So I'm going to be telling you all about the work that I've done over the past 4 years at the Koch Institute in the Sánchez-Rivera lab, and I've titled my thesis, Looking Backward and Forward at Cancer-Associated Mutations with Precision Genome Editing. So I wanted to start this presentation with a question, and that is, how do the mutations that we observe in cancer patients determine the behaviors of their cancer? And given that this is a broader audience, I wanted to take the time to actually build up from first principles so everyone understands both why this is an important question and appreciates some of the techniques and approaches that we've developed to break off and answer small pieces of this question. So first, fundamentally, cancer is a genetic disease that's driven by diverse genetic alterations. What I'm showing you here is a karyotype of a normal human cell. So karyotyping is a method of visualizing the chromosomes that are present in a single cell. And what you'll notice is that there's 2 copies of each of your 23 chromosomes, one from your mother, one from your father. Whereas if we look at an example tumor cell, you'll notice that things are all messed up. So you can have duplications or deletions of entire chromosomes, as well as the shuffling of chromosome arms. So for example, a piece of chromosome 9 could be attached to a piece of chromosome 3. And these large-scale chromosome level alterations are very, very common in cancer patients. These are called copy number alterations. But in addition, what we observe in cancer patients are tens of thousands or hundreds of thousands of single nucleotide variants. That is, single letter changes to the genetic code. And these are really going to be the focus of the work that I'll be telling you about today. So we can see this when we look at this sequencing of a single patient's lung tumor, where each of these red dots represents a single nucleotide variant. And you can see that there are tens of thousands of these that are scattered throughout the genome of this patient. And we can look more broadly at a cohort of around 2,500 patients, and we can identify tens of millions of mutations in this patient cohort. And this does vary as a function of cancer type. So for example, lung and skin cancer have the highest mutational burdens. But in general, we can expect to observe on the order of tens of thousands of unique single nucleotide variants in a given cancer patient. So you might be wondering, why are these mutations important? How do they actually exert their effects? And so to understand this, we first need to understand how the information that's encoded in DNA is actually used by the cell. So this is what's known as the central dogma of molecular biology. And so essentially, we have our genome, we have our DNA. And so there are around 20,000 genes in your genome. And when a gene is turned on, it's transcribed into a piece of RNA, a transcript of RNA, which is in turn translated into a string of amino acids that fold into a protein. And so proteins are the actual functional molecular machines that perform all of the operations that are necessary for your cell to survive. And just to provide us a sense of scale here, a typical protein is in the order of a few dozen to a few hundred atoms across in terms of dimensions. So we are working at the molecular level here. And really, there's an astounding diversity to the structures that proteins can take, and these unique structures allow them to perform their particular function in the cell. So for example, there are proteins that are involved in transcribing genes, so turning DNA into RNA, sensing stress in cells, transporting molecular cargo, and synthesizing ATP. So when a mutation occurs in DNA within a gene as a result of, for example, an error in replication or some sort of carcinogen, this in turn results in a mutation in the RNA transcript and can produce a mutant protein. And so this mutation in this protein, this change in the amino acid sequence, can alter the function of that protein. For example, it could change the ability of this protein to sense stress in cells. So what occurs in cancer cells and in cancer patients is that you have sets of mutations that work together to drive the malignant phenotypes, the uncontrollable cell growth that actually brings about cancer. So really, what makes cancer so difficult to treat and understand is that every cancer patient is unique and their tumors are dynamic. So cancers can occur in different organs and in different cell types. Even if we look within a single tumor, there are dozens of different cell types. So there are the cancer cells themselves, there's a zoo of different immune cells, there's blood vessels, neuronal cells, as well as normal stromal cells. And even if we focus on only the cancer cells that are present in that tumor, there's multiple levels of variation. So for starters, everyone has a unique genome that can vary by millions of base pairs from the person sitting next to you. And on top of this, cancer cells acquire sets of tens of thousands of mutations that are unique to each cancer patient. And even for two cancer cells that are genetically identical, there can be epigenetic differences. That is, they can express different sets of genes that imbue them with distinctive properties. And on top of this, we're trying to hit a moving target. So cancer cells continue to evolve as cancer progresses. So really, this changes continually over time. And however, despite this extensive diversity that we see in cancer patients, the mutations that we see in patients really aren't random, but rather are united by a shared molecular logic. So even though the exact variants that we're seeing in patients may differ, at the end of the day, their cancers need to activate pro-growth signals while inactivating anti-growth signals. And in molecular biology parlance, we call this the activation of oncogenes and the loss of tumor suppressor genes. And a decent analogy here is you can think of this like losing the brakes with the loss of a tumor suppressor gene and stepping on the gas with the activation of an oncogene. So back to this original question that I posed at the start of this presentation. How do the mutations that we observe in cancer patients determine the behaviors of their cancer? And really, I think Richard Feynman has a nice quote, which I think sort of encapsulates our approach here, which is, "What I cannot create, I do not understand." He has another quote, which I quite like, which is, "Never confuse an education with intelligence. You can have a PhD and still be an idiot." So, good reminder for my friends and family. But within this overall framework, to understand cancer, we must create cancer. In order to understand the effects of a particular mutation, we have to create that mutation. So within this framework, the overall goal of my work has been to engineer cancer-associated mutations to better understand their underlying biology and inform precision oncology. And I think we can really think about this in two ways. We can look backward at mutations that we've observed in patients that arise as the result of different selective pressures. So for example, exposure to different cancer drugs, the effects of the immune system, metabolic pressures, as well as oxidative stress, among many other different selective pressures. But we can also look forward at mutations that we haven't yet seen in patients, but that we can expect might arise as a result of, for example, novel drugs or different selective pressures. And in both of these cases, my work employs high-throughput variant engineering to create thousands of these mutations in parallel, to hopefully be able to functionally screen them in diverse contexts, so in the context of these different selective pressures, to better understand how these mutations function and hopefully enable actionable discoveries. So I'm going to be telling you about two stories today. The first looks backward to functionally characterized over 1,000 clinically observed mutations in p53, which is the most commonly mutated gene in cancer. And the second story will be predicting mechanisms, looking forward to predict mechanisms of resistance within the cyclin-dependent kinase family, or the CDK family. But really, for both of these stories to make sense, we first need to understand, what do I mean by high-throughput variant engineering? So to engineer and functionally characterize and test thousands of mutations in parallel, we're employing CRISPR-based gene editing tools. So the way that CRISPR works, there's two components to this system. So there's a protein, Cas9, which essentially acts like a pair of molecular scissors, creates a double-stranded break at a defined location in the genome. And Cas9 takes direction from the information that's encoded in a guide RNA. So these guide RNAs contain a protospacer, which is a 20 nucleotide sequence that has homology or matches with a location in the genome. And you can think of this like an address in Google Maps, where the guide RNA is specifically defining a location in the genome. And this is, you actually have enough specificity in a 20 nucleotide sequence. There's actually over 1 trillion possible unique 20 nucleotide sequences. Our genome is only 3 billion base pairs, so this is enough information to precisely target one spot in the genome. So the way that this works, the guide RNA will bind to Cas9. This will search throughout the genome until it finds its matching address, so its matching protospacer in the genome, at which point Cas9 will actually unwind DNA and create a double-stranded break. And generally, this double-stranded break is repaired imperfectly by cells, so it results in the insertion or deletion of a random number of nucleotides. And if this double-stranded break is located within a gene, it generally results in the loss of a protein. So really, the power of CRISPR is that it's high throughput. So what do I actually mean by this? What we're able to do is we can deliver a virus pool with guide RNAs targeting different genes, so that has different addresses in the genome, to a population of cells that express Cas9. And what we end up with is a population of cells that essentially have different genetic knockouts. We can then expose these cells to different selective pressures, for example, different drugs. And in this way, we can understand which of these genetic knockouts confers a particular selective advantage. And the way that we're able to do this is essentially by sequencing at the start and the end of the experiment to count these guide RNAs. So in this particular example, these blue cells, which received guide RNA Y, seem to predominate in this population. They seem to have a selective advantage, so that we can say that knockout of gene Y, as programmed by guide RNA Y, confers a selective advantage in this case. And so the issue with traditional CRISPR-Cas9 is that it knocks out genes, so it results in the loss of a protein. However, most of the cancer-associated mutations that we actually see in patients are single nucleotide changes that actually result in a mutated protein. So they actually modify an amino acid as opposed to fully knocking out that gene. So in order to actually generate these single nucleotide variants, we need to modify the CRISPR architecture slightly. So we're still using our guide RNAs to bring this editing machinery to a particular spot in the genome. But we have to modify Cas9 so it's no longer making double-stranded breaks. And what we do is we can tether other proteins to Cas9. And these other proteins are actually able to generate these single nucleotide variants. So I'm going to be telling you about two technologies, or these projects have used two technologies that I'll be showing you. The first is base editing, which allows you to make C to T and A to G changes in DNA. And the second technology is prime editing, which essentially allows you to make any small mutation anywhere in the genome. And so the first project that I'm going to be discussing really focuses on the use of prime editing. So let's dive in a little deeper to see how this actually works. So most of this should look familiar based on what I just showed you. You still have your protospacer sequence, which defines an address, a location in the genome. And we still have our Cas9. In this case, it's a nicking Cas9 that makes a single-stranded break here. What's unique about prime editing is that our prime editing guide RNA, or our PEG RNA, encodes a replacement sequence. So this is search and replace genome editing. So the way that this works is that this replacement sequence actually encodes the mutation that you're trying to engineer. And so this replacement sequence catalyzes the synthesis of a single-stranded DNA by the reverse transcriptase that's tethered to our nicking Cas9. And then with some probability, this strand of DNA that contains our edit of interest is actually incorporated into the genome to very precisely generate our mutation that we're trying to engineer. And so, prime editing is a really transformative technology. This was developed by David Liu's lab back in 2019, and really, the prospect of working with this system is one of the things that drew me to Francisco's lab, because frankly, it sounds like science fiction. It's a level of control over the genome that really I think geneticists have dreamed about since the discovery of the structure of DNA. In theory, it allows you to make virtually any small mutation pretty much anywhere in the genome. The issue is that it's limited by variable or low editing efficiency. And one of the factors that contributes to this variable editing efficiency is that designing PEG RNAs is actually quite difficult. And one of the reasons for this is that replacement sequence that I was talking about actually contains two subsequences that can both vary in length. So as a result, for a given mutation that you're trying to engineer, there can be hundreds of possible PEG RNA designs. And it's very likely that only one or a small subset of those will actually edit with high efficiency. And so we wanted to use this tool to actually screen and test thousands of mutations in parallel. And so we asked, how do we actually overcome this challenge of lower variable editing efficiency to enable screening with prime editing? And so we think the answer lies in the prime editing sensor architecture, which links pegRNA identity to editing outcomes. The way that this works is we have a DNA construct where we have our pegRNA, and then right next to it, we have a synthetic copy of the endogenous target site. So our pegRNA is transcribed, it binds to the prime editor protein complex, and then this complex actually edits at both our sensor site and the endogenous target site. And the reason this is important is because we can sequence this entire construct, and we can define both the identity of each pegRNA, but also these editing outcomes at this sensor locus, which correlate strongly with what happens endogenously. And so really, this is important because it allows us to identify optimal pegRNA designs. We can design multiple pegRNAs for each mutation that we want to engineer and select only the ones that are highly efficient based on their editing at the sensor locus. But it also allows us to filter screening data to remove low efficiency prime editing GAD RNAs. So we wanted to use this approach to screen thousands of cancer-associated mutations in parallel. And as a prototype, we wanted to test mutations in P53, which is the most commonly mutated gene in cancer. So P53 is a protein that essentially senses stress in cells. So it can sense DNA damage, oncogene signaling, hypoxia, among many other stresses, and it produces a cellular and transcriptional response. Most famously, it can induce cell cycle arrest, so it can stop cells from actually growing and dividing, or it can actually tell cells to commit suicide in a process that's known as apoptosis. And so the way that P53 actually does this is by acting as a transcription factor. So if we look at this structure of P53 here, actually 4 P53 proteins come together to form a tetramer complex that then binds to DNA. And this DNA binding at specific sequences can activate the transcription of other genes, which in turn provide this response to one of these stressors, for example, turning on genes that result in apoptosis. And so based on this description, you can probably understand why there's such a selective pressure for cancer cells to inactivate P53. They probably don't want to kill themselves if they want to persist as a tumor. And so this actually is the case. So P53 is mutated in around 50% of all cancers. What I'm showing you here is mutations at each amino acid position or at each codon in P53, and this is among a cohort of around 100,000 patients. And what's pretty striking here is that there are some clear amino acids or codons where mutations are much more frequent in P53. We call these hotspot variants, and there's around 7 of them. And these all occur in the DNA binding domain of P53, so they actually alter the ability of P53 to interact with DNA and activate the transcription of other genes. But if we zoom in on this plot here, what you'll notice is that there's actually frequent mutations throughout the entire sequence of P53. So these hotspot variants, these 7 variants that I'm highlighting, are the most well studied, and they represent around 25% of the observed variation in P53. But what about this remaining 75%? So no one previously had actually systematically characterized all of these variants that we observe in patients, but really this 75% represents millions of patients every year because these P53 variants are so common. And so what we wanted to do is we wanted to systematically characterize and evaluate these mutations that we see throughout the entire sequence of P53 with prime editing. We asked the question, are all these mutations created equal, or do they have different functional consequences? And we also wanted to see if our sensor system worked. So we wanted to see if we could use this system to actually identify efficient pegRNAs by designing around 30 pegRNAs per mutation. So for starters, we had to design a prime editing sensor library, so our pegRNA sensor library. So for this task, I took data from around 40,000 patients, and I selected the recurrently observed variants in P53, of which there are around 1,000. And then to actually build this prime editing sensor library, I actually had to design a Python package that I called PEG, or Prime Editing Guide Generator. So I'm not going to get into the nitty-gritty details about how this works, but this was a big learning journey for me, something I'm very proud of. Essentially what it does is it takes in these input variants and it automatically produces these pegRNA sensor constructs, so it automates this process of pegRNA design. And what's nice is that since this is published as a Python package, essentially in a matter of minutes, anyone with a computer can, in theory, generate a prime editing sensor library. So it really sort of democratizes this technology. So PEG generated a library of around 30,000 pegRNA sensor constructs. So around 30 pegRNAs designed for each variant, so scanning that pegRNA design space to try to identify the most efficient pegs. And we can visualize what this library actually looks like. So essentially, we're trying to engineer variants throughout the entire sequence of p53. And so the way that these experiments actually work, we deliver this prime editing sensor library as a virus to lung adenocarcinoma cells that express our prime editor protein. And so we then split these after we allow a few days for editing to occur. We split this into two conditions. One is untreated, and one is a Nutlin treatment condition. And so Nutlin is a small molecule that essentially activates p53 in cells. So this is a way of actually selecting for cells that have inactivated p53. And so what we're able to do is at various time points over this around month that we screened these p53 variants, we can extract genomic DNA, and we can sequence our pegRNA sensor construct to both look at the enrichment and depletion of these pegRNAs and also our editing outcomes at our sensor locus. So diving right into the data, what I'm showing you here is the average correct editing efficiency at our sensor locuses for all the pegRNAs in our library, if we look at this plot to our left, at each of the time points and conditions that we characterized. One thing you can note is that it does seem that editing seems to continue to increase over time, and this seems to level out at around a correct editing efficiency rate of 8% if we consider all pegRNAs. If we consider only the most efficient pegRNA for each variant, this nearly doubles to editing efficiency of around 14%. So we can already see that our sensor is working. It's allowing us to select more efficient pegRNAs. But really, these averages don't tell the full story. So this is about as far from a normal distribution as you can get. It's much closer to an exponential distribution, where essentially, if we look at most of our pegRNAs have essentially zero editing activity. So most of our pegRNAs aren't working at all. And then we have this long tail where some of our pegRNAs are working with very, very high efficiency, so up to even 95%. So the first question that we asked is, what determines the efficiency of a pegRNA? And I'm not going to spend too much time on this, but in short, we were able to identify and recapitulate many features that predict the efficiency of pegRNAs. One that I think is particularly interesting is actually the location of your mutation of interest in that replacement sequence is quite important. So it seems that if you install your variant to actually disrupt your protospacer or your PAM sequence, that leads to a big boost in editing efficiency, likely because it prevents your prime editor from actually rebinding to your locus that you're trying to engineer. But we were also able to generate a simple machine learning model that reasonably predicts prime editing efficiency, and I've actually incorporated that back into the Python package that I developed. But really what we were more interested in is how do we actually, can we interpret this noisy data to gain biological insights? So can we control for this variable editing efficiency to gain some biological insight? And this is where the prime editing sensor really shines, is because we have an empirical measurement of the activity of each pegRNA, we can use this to actually filter out pegRNAs that are low efficiency to really boost our signal-to-noise ratio. And so this is what the data actually looks like. So each of these dots represents a different pegRNA. The x-axis is the amino acid position or the codon where the pegRNA is trying to engineer a mutation. And the y-axis is essentially showing the fitness of these cells that get these pegRNAs. So things that are higher up tend to have a selective advantage. So in this case, they seem to grow and survive well in the presence of Nutlin, and things that are lower down tend to die at a higher rate or grow more slowly. So really, this data is quite noisy. There's no clear structure in this data, but what we can do is we can filter out our low efficiency pegRNAs. And what we see is we see some clear structure starting to emerge, where we have some variants that are clearly enriching and depleting at certain regions in this p53 protein, and we can really choose any editing filtration cutoff here. So I'm showing you 20% editing cutoff. We can also boost this up to a 40% editing cutoff. And so we think that this effectively mitigates the problem of variable editing efficiency in using prime editing in high throughput because we have an integrated measurement of the activity of each of these pegRNAs. So I mentioned earlier that essentially we can count guides at the start and the end of an experiment to determine the selective advantage that's provided by a cell receiving a particular guide RNA or pegRNA. What we can also do is count mutations, which is actually sort of the gold standard approach. So we can just sequence the endogenous locus, count mutations at the start and the end of an experiment to look at the selective advantage that's provided by a given mutant. And if the efficiency of editing were 100%, so if each pegRNA edited with 100% efficiency, these things would actually be perfectly correlated. But we know that that isn't the case. We see a pretty wide and high variance distribution of prime editing efficiency. So we went ahead and actually did this experiment. So we sequenced the endogenous p53 locus as opposed to just relying on these prime editing sensor constructs. And we compared the behavior of these mutations with the pegRNAs that we sequenced. And so if we look on the bottom right here, if we don't filter out any pegRNAs, there's essentially no correlation between the behavior of, or the count of these mutations and the count of these pegRNAs. Whereas when we increasingly filter out low-efficiency pegRNAs, we see that there's a clear correlation that starts to emerge. And this is particularly important because it tells us that if you want to use prime editing in high throughput, essentially, without a prime editing sensor, you would have to sequence every single locus that you're engineering, whereas with our system, we can, in theory, mutagenize thousands of different sites while controlling for the editing activity of each pegRNA. So it's a much more powerful approach. So we also validated some of the phenotypes that we saw induced by these variants with competition assays. So I selected 29 pegRNAs for follow-up validation. These were spread throughout the sequence of p53. They had a range of fitness effects and a range of editing activities. And so we performed competition assays where we deliver these pegRNAs individually. And what we do is we co-deliver in this construct a fluorophore, in this case RFP, that turns these cells red. So every cell that receives a pegRNA turns red, and then we can mix this back with the parental uncolored population. And what we're able to do is essentially we can track the fraction of that cell population that's red to quantify the fitness effects of a cell receiving a certain pegRNA and engineering a certain mutation. So this is what this data actually looks like. These are two different variants that both strongly enrich in the presence of nutlin. So you can see that, essentially at the end of this 2-week period, all of these cells are red because all of these pegRNA receiving cells have been enriched. We can also use this data to look at how well our sensor is predicting endogenous editing because we're transducing these things individually. And as a nice sanity check, we do see that there's a good correlation between sensor editing and endogenous editing activity, which is nice to see. Particularly for missense variants, there seems to be more noise for our insertions and deletions. But then we went ahead and actually performed all 29 of these competition assays, and we saw there was a good correlation between the behavior of these pegRNAs, these mutations in our screen, and when we test these things individually. And actually, this correlation seems to increase when we filter out our low-efficiency pegRNAs. So one thing that I haven't told you is that there's actually more simple ways to test individual mutant genes, individual mutant proteins. So what we're able to do is we can deliver a cDNA construct of a gene that has a mutation of interest. The issue is that in general, you'll express very high levels of your protein of interest, whereas when we engineer mutations endogenously, for example, a gene may only be turned on or transcribed and translated in the presence of a particular cellular and molecular stimulus. And there are also complex feedback loops that control the precise levels of a protein in a cell. So this is sort of akin to studying the behavior of a chimpanzee in a zoo as opposed to in its natural habitat in the jungle. You might end up with some artificial results if you only look at how this chimp behaves in the zoo. So what was nice is that there was some previous work that had actually used this more artificial approach, this cDNA-based overexpression approach, to actually test all of the possible single amino acid variants in p53. So every small amino acid variant possible in this protein. And this was actually performed in the same cell line, in the same nutlin treatment condition. And so what this allowed us to do is actually to test the behavior, or to compare the behavior of these p53 variants in both of these systems, so when engineered endogenously and when overexpressed. And the first thing that's quite striking, that was pretty surprising to us, is that there's actually very little correlation between the behavior of mutations in each of these systems. And in particular, when we break this down into the different functional domains of p53, so p53 is a protein that essentially has different domains that perform different functions. This difference was particularly pronounced in mutations that were located in the oligomerization domain, or the OD. So that's what's shown by this red box plot here, where what we saw is that when we engineer mutations in this OD endogenously, we see them enrich quite strongly, whereas when they're overexpressed with this more artificial approach, these mutations don't seem to have a fitness effect or a fitness advantage. And so this domain, this oligomerization domain, is actually involved in the tetramerization of p53. So as I mentioned earlier, 4 p53 proteins have to come together to form an active complex that can bind to DNA and transcribe genes. And so the OD is actually what performs that that tetramerization. And so we see essentially, it seems that we're only seeing these fitness-based effects of mutations in this OD when we're engineering these things endogenously. So this was quite interesting to us. And so we wanted to test this further. So we went back to our competition assays, but this time we also tested those cDNAs. First thing that was nice to see as a sanity check is that these cDNAs behaved similarly in our own hands as they did in that paper that we took them from. But when we compared the behavior of mutations in the OD that were delivered with a cDNA or engineered endogenously with prime editing, what was quite striking is that we validated that these variants only enrich when engineered endogenously, so they only cause a fitness advantage in cells when we engineer these things endogenously, but not when they're overexpressed. And this was quite exciting to us because it tells us that really gene editing is important in this context. We're only going to be able to see the effects of some of these mutations if we actually create them in their natural context. Also, when we looked in the literature, we found that many of these OD variants are actually frequently observed in Li-Fraumeni syndrome patients. So these are patients that have germline mutations in p53 that predispose them to higher rates of cancer. And in addition, it seems there was some other work that has shown that these form stable dimers instead of tetramers that could result in some neomorphic p53 functions. So these are some nice sort of external validations that what we're seeing does seem to be real. So in conclusion, I've hopefully showed you that our prime editing sensor approach allows us to screen mutations with prime editing in high throughput and filter out low efficiency pegRNAs to boost our signal to noise ratio. We can also use these data sets to identify the features of efficient pegRNAs and build simple machine learning models. And also using this approach, we can find some phenotypes that are otherwise concealed by more artificial cDNA-based overexpression systems. So in addition, we've also extended the prime editing sensor approach to characterize different genes or mutations in different genes. In particular, we characterize variants in KEAP1, which is another tumor suppressor gene. So I don't have time to go into too much detail here, but this was a nice story where we showed that multiple KEAP1 variants are of unknown significance, which were associated with poor clinical outcomes in lung cancer, or actually loss of function. And so we used a nice transcriptional reporter system here. And what's also been nice to see is that there have been multiple other groups that have made use of this prime editing sensor approach. So it's always nice to see the adoption of your approach by the broader community. And so that brings me to the end of the first part of my talk, where we functionally characterized over 1,000 clinically observed mutations in p53. It's also a good opportunity to grab a sip of water. And so that brings me to the second part of my talk, where instead we look forward, in this case, to try to predict mechanisms of resistance to the cyclin-dependent kinase or CDK family of inhibitors. So just to jump right in here, the problem that we're trying to address is that targeted cancer therapies are initially effective, but tumors eventually acquire resistance. And one very common mechanism of resistance is actually mutation of the drug target itself. So the way that most modern cancer drugs work, most modern targeted therapies, is that drugs are designed to bind to and inhibit specific proteins in cancer cells to stop the growth of those cancer cells. But what can happen is that cancer cells can acquire mutations in the actual protein targets, so in the genes that produce the proteins that are being targeted by these drugs, that render those drugs ineffective. So they, for example, no longer bind to their target proteins. And so cells become resistant, they don't respond to those drugs. And so there's many examples of this across many protein inhibitor combinations. So for example, EGFR, as well as BCR-ABL. And what we even see in patients is that patients that are treated with a first-line therapy can develop resistance to that therapy. And then if they're treated with a second-line therapy, they can actually sequentially acquire additional variants that render that second-line therapy ineffective. So it's sort of like a game of cancer whack-a-mole. And generally, these resistance mutations are first identified in the clinic, and we asked, can we prospectively identify resistance variants before drugs are given to patients? So the focus of this project is the cyclin-dependent kinase family, or the CDK family. So this is a family of around 20 proteins, CDK1 through CDK20, not a very creative naming scheme. And this is actually part of a larger family of proteins known as kinases, of which there are around 500 in the human genome. This is actually one of the largest protein families in the genome. So what kinases do is they phosphorylate things. So a kinase will bind to ATP, it'll bind to its substrate protein, and it'll take one of the phosphate groups off of ATP and attach it to the substrate protein. And so this is one of the most ubiquitous methods that proteins use to signal one another in the cell. And so, for example, this phosphorylation tag could activate this protein and cause it to signal downstream to promote cell growth, for example. So this is what the actual structure of CDK2 looks like. One additional complication with the CDK family is that they have a cyclin binding partner. So this is just another protein that they need to bind to actually achieve an activated form. And so what you see here is that this CDK2 protein is hugging this ATP molecule. So it's sort of sandwiched between the two lobes of this protein. So what actually occurs is that a substrate peptide, which would be part of a larger protein, fits into this cleft, and then one of the phosphate groups from ATP is actually transferred onto this substrate peptide. And so there are actually two classes of CDKs. The first are the cell cycle CDKs. So these are CDKs that actually phosphorylate other proteins to control the cell cycle, and the cell cycle is essentially the method by which cells control their replication and division. And CDK4/6 inhibitors, which are CDK4 and CDK6, are both cell cycle CDKs. CDK4/6 inhibitors have been very efficacious in breast cancer, in particular in combination with hormone therapies. So you can see this if we look at this clinical trial data. If we look at that red line where we're plotting progression-free survival, essentially there's been a marked survival improvement for patients that are treated with these CDK4/6 inhibitors. However, the issue is that on the order of around 70% of women treated with these inhibitors will actually develop resistance to the therapy, or these inhibitors might not work at all when they're first treated. And we understand some of the genetic underpinnings in terms of what causes resistance to these therapies, but in general, in the majority of cases, we actually don't understand what causes resistance. In addition, there's another class of CDKs, another sort of subfamily of the CDK family, and these are the transcriptional CDKs, which coordinate a different cycle. In this case, it's the transcription cycle. And so the transcription cycle is actually the cycle that controls, you might have guessed it, how genes are transcribed. So the details aren't super important here, but essentially most of the action of these kinases focuses on the C-terminal tail of RNA polymerase II. So this is a very well-orchestrated process where RNA polymerase II binds, and then the C-terminal tail is phosphorylated in sequence by each of these CDKs to allow it to actually transcribe genes. And so the development and testing of transcriptional CDK inhibitors is a very active area in oncology. I'm showing you a table here of some of the ongoing clinical trials. And really, these are licensed for many diverse cancers. And what's quite interesting is actually that many of these transcriptional CDKs are essential genes, so they're essential to the activity and survival of each of the cells in your body. But it does seem like there's a therapeutic window where we can effectively inhibit these things, because it seems that cancer cells rely on sort of oncogenic transcriptional programs more so than normal cells. So there is a window where we can effectively dose and inhibit these CDKs. So just to summarize, resistance remains a major challenge with CDK4/6 inhibitors despite their clinical success, and resistance to transcriptional CDK inhibitors will likely present a challenge to clinical success, but it hasn't been systematically explored because these drugs are in clinical trials. And really, to overcome resistance to targeted therapies, we must first identify mechanisms of resistance. So, the gold standard method of identifying mutations that cause resistance to a certain therapy relies on natural variation in a population of cells. So you take a large population of cells, say on the order of 100 million cells, and over the course of many months, you can increase the dose of your targeted therapy of interest until hopefully you can isolate and expand a resistant clone that is now resistant to your therapy of interest. You can subject that to sequencing and hopefully identify a mutation that causes resistance to that therapy. And just to provide some numbers here, as I mentioned, you would generally have to treat on the order of 100 million cells to just be able to identify one cell, one clone, that has a mutation that causes resistance to your therapy. So this is a very time and labor-intensive approach, and it doesn't really allow you to directly compare the resistance profiles of different drugs, for example. So instead, our approach is to use essentially directed evolution. So we're using base editing to specifically mutagenize these genes that are producing proteins that are being inhibited by these different drugs to essentially speed up this process. So what we're able to do, what I'm showing you here, each of these blue bars represents a protospacer, so an address in the genome where we're bringing, in this case, base editing machinery. And so what we're doing is we can specifically mutagenize these genes that are producing proteins that are the targets of these drugs to sort of speed up the process by which we can identify these resistance mutations. So essentially what I've done is I've generated as many possible protospacers as I can to tile this gene to maximally mutagenize it, so to maximally explore that mutational space. And so the way that this works, we have our tiling base editing guide RNA libraries. We can deliver this to either adenine or cytosine base editor expressing cells to produce A to G or C to T edits. After we allow editing to occur, we can actually treat with drug and use this to identify resistance mutations. And this works because we have our CDK drug pairs in this case. And essentially, we're generating mutations in cells in these CDK proteins through directed evolution. And when we treat with drug, only the cells that actually are resistant to these therapies will actually survive, so we can use this to quickly identify mutations that cause resistance. So I generated base editing tiling libraries for all of the major transcriptional CDKs as well as CDK246. And these are actually broken into 3 separate sub-libraries that I screened independently. And so these are loosely broken up based on function. The first of these contains CDK7, 8, 9, and 19. So these are proteins that are involved in the first steps of the transcription cycle. The second subpool contains CDK12 and 13. So these are transcriptional CDKs involved in the latter steps of the transcription cycle. And then subpool 3 contains CDK246, which are all cell cycle CDKs. So we can deliver these libraries independently to either adenine or cytosine base editor expressing cells. We can allow editing to occur, and then we can treat with one of these 15 different CDK targeting therapies. So just to summarize, essentially we're mutagenizing these CDKs, we're treating with drug, and we're identifying resistance variants on the other side. And so one thing to highlight is the fact that we were able to test 15 different CDK targeting therapies, and these experiments really only take a matter of like 1 month. So we treat with drug for a period of 3 weeks. Really speaks to just the power of this approach to very quickly identify resistance variants, as you'll see. So one other sort of additional technical piece here is that we're testing drugs that have diverse mechanisms of action. So this includes inhibitors, PROTACs, and molecular glue degraders. So inhibitors are small molecules that essentially just sit in a protein and inhibit its function, whereas PROTACs and molecular glues essentially recruit other proteins, ligases, that deposit ubiquitin tags on your target protein, in this case, one of the CDK proteins, that tell that protein to be degraded by the cell. So they actually cause that protein to be degraded by the cell. So what's nice is, since our approach, essentially, we can test many different drugs that have different mechanisms of action, and we can compare the resistance profiles that we identify among these different drugs, even if they're targeting the same protein, which is quite exciting. And once again, we're using our sensor system. In this case, it's the base editing sensor system. Works very similarly. So we can sequence this entire cassette, and we can get these editing outcomes at the sensor locus. And the reason that this is particularly important here is that we're actually not defining a set of mutations that we want to engineer a priori or before our experiments. So we're just generating as many protospaces as we can, tiling these genes. So what the sensor allows us to do, it actually tells us the activity of each of these guide RNAs, because we don't have a specific set of variants in mind. And I've written some code that actually translates this all the way down to the protein level, so we can understand what's the sort of amino acid effect, or the amino acid changes that are being induced by each guide RNA. And so this is an example of what this data actually looks like. This is real sensor editing data. So what I'm showing you at the top is the sensor editing at the DNA level. So those blue T's have been converted from C's into T's. And just to note, the editing efficiency of this is very, very high, and we can see the effect of that at the protein level in red, where this is producing an L to F mutation at the amino acid level with very high efficiency. So the first question we asked is sort of what fraction of the mutational space are we actually able to explore with these libraries? And there's really 2 ways to quantify this. The first is depth. That is, at each amino acid position in each of these proteins, what fraction of the 20 possible amino acid substitutions are we actually observing? And the answer is that we usually observe around 1 to 3 amino acid substitutions at each residue. This is just based on the limitations of the types of edits that are being made by our base editors. But really where this approach shines is if we consider instead the breadth of mutagenesis. That is, if we look at these protein sequences, what fraction of all the amino acids in these proteins are being mutagenized? And essentially, even at a very high sensor editing cutoff of, say, 50% editing, we can still see that around 80+% of the amino acids in each of these proteins are being mutated. So essentially, we can almost mutate every amino acid in each of these CDKs. And so this is a trade-off that you have to make when you're choosing your gene editing technology, but we think that this is the correct trade-off in this particular case, and hopefully our data will show that in the following slides. So I mentioned earlier that the gold standard method of identifying resistance variants is this dose escalation approach that relies on natural variation in a population of cells. And so we performed many of these screens, and what was quite exciting to us is actually that 3 of the compounds that we tested, previous groups had actually used these gold standard dose escalation methods to identify resistance variants. And in all 3 of these cases, the mutation that they identified matched with some of the top hits from our directed evolution base editing screen, which was, sort of the perfect orthogonal validation that we're identifying genuine bona fide resistance variants. So the first example of this is with a CDK9 targeting PROTAC, where this group identified the CDK9 L156F, so leucine 156 to phenylalanine mutation, as conferring resistance with this dose escalation approach. We identified the same variant. So I'm going to be showing a decent number of these plots. So just to clarify, each dot is a guide RNA. The x-axis is the location of the mutation that it's engineering in each of these protein sequences, so CDK7, 8, 19, or 9 in this case. And the y-axis is essentially the resistance score. So things that are higher up confer resistance to a therapy. Things that are lower down actually sensitize cells to a therapy. And so in this case, we had 4 guide RNAs that all engineer our L156F mutation with very high efficiency. And these were strongly enriched in the presence of this CDK9 targeting therapy, matching these sort of gold standard approaches. We see the same thing if we look at a CDK12 targeting molecular glue, where we see the same resistance variant popping up, as well as a CDK12 targeting PROTAC, so a slightly different modality of therapy targeting CDK12. And so one thing that's very important to highlight here is while these approaches identified 1 or 2 resistance variants in response to each of these therapies, we can essentially mutate, since we're mutating every amino acid in each of these CDKs, we can identify dozens and dozens of resistance variants in a much shorter period of time. And it also allows us to directly compare the resistance profiles of each of these inhibitors, so it's a much more powerful approach. So we performed many of these screens. I don't have time to go dive into all the data, but I do want to sort of just highlight what I think are some of the most biologically interesting results. The first is that we identified and validated the first ever resistance variant in CDK7, which is the L18F variant. I sometimes call this the Gould variant if I'm feeling myself, but I think I'm the only one. So we actually have 3 guide RNAs that all engineer this L18F variant with high efficiency, and these were strongly enriched in the presence of our CDK7 targeting therapy. We can actually map these to the structure of CDK7. We can see that they're located right in the ATP binding pocket where this drug would be binding, so they're likely altering the affinity of this molecule. And these validate strongly when we test these individually. So we can actually select for a homozygous CDK7 population, and if we do dose response curves, we can see a very strong resistance phenotype being mediated by this mutation. In addition, we identified other variants in CDK9 beyond L156F, and many of these map again to the ATP binding site. What was quite interesting to us, actually, is so we tested 2 different CDK9 targeting therapies. One was an inhibitor, KB0742, and one was a PROTAC. But the actual CDK9 binding part of these drugs was quite similar. But when we tested L156F, that CDK9 resistance variant, what we found is that this mutation only caused resistance to the CDK9 targeting PROTAC, but not this inhibitor. And it actually seemed that it slightly conferred increased sensitivity to the CDK9 targeting inhibitor. And this is something that I'm going to come back to in the following slide. But we were also able to perform RNA sequencing on CDK mutant and wild type cells. And we can expose these things to drug, and essentially what's quite clear is that there's essentially no transcriptional changes for our resistant cells when we expose them to drugs, so they really don't care when we expose them to drug, whereas there's quite dramatic transcriptional changes for our wild-type cells. And there are also some basal differences between our resistant wild-type cells at baseline. So coming back to this observation that we have a mutation that causes resistance to one CDK9 targeting therapy but not another, really this sort of gave us the opportunity to mirror the sequential acquisition of mutations that I discussed earlier that we see in patients. So we often see in patients is they're treated with one targeted therapy, they develop resistance to that drug, and then they develop additional mutations that render a second drug that they're treated with ineffective. And so given that we have this L156F variant that causes resistance to this CDK9 targeting PROTAC, but not the inhibitor, we asked, can we identify additional mutations, for example, in CDK9 that might confer resistance to this therapy, sort of mirroring the first and then second line therapy approach that we see often in patients. And so to do this, we first generated a homozygous population of L156F cells, so CDK9 L156F cells, by selecting with our drug. And really what's unique about this approach is that it's iterative. So we can actually just deliver this library back to these cells to mutagenize them in this CDK9 L156F genetic background. we can allow editing to occur, and then we can treat with the CDK9 targeting therapy to identify resistance mutations on this genetic background. And what was quite surprising to us, quite unexpected, is that we actually observed enrichment for many CDK7 mutations. So this is surprising because this drug is targeting CDK9. But when we actually look back at the original paper that developed this compound, this was from Angela Kohler's lab at the KI, we found that CDK7 is actually the top off-target for this therapy. So what we think is likely happening is that this drug, at the concentrations that we were testing it, is likely binding and inhibiting to both CDK7 and CDK9. So you need resistance mutations in each of these proteins in order to develop resistance to this therapy. So we actually tested this individually. We made a CDK9, CDK7 double mutant. And so if we look at this purple line, essentially we see that this still retains resistance to a CDK7 targeting therapy. It's still resistant to our CDK9 targeting PROTAC. And while there isn't a dramatic resistance phenotype, it does seem that it sort of brings that hypersensitivity of L156F back to baseline. So I think this really shows the power and flexibility of this approach to sort of match the mutation acquisition process that we see in patients. So up until this point, all of the experiments that I've showed you have been with cells in a dish. So we wondered, are these resistance mutations that we're identifying, do they confer resistance in the more physiologically relevant environment in a mouse? So we actually engineered these CDK9 and CDK7 resistance variants in murine or mouse leukemia cells. And so then we observed strong selection for these CDK7 and CDK9 resistance variants in these mouse cells when we treated with these drugs. And what we were able to do is we can inject these cells back into the mouse, and we can either let them grow or we can treat with one of these CDK9 and CDK7 targeting therapies and test whether they cause resistance. And actually what was most striking to us, if we compare these two gray bars, the non-treated conditions, what I'm showing you here is the fraction of leukemia cells in the spleen. And what was quite striking is actually it seems that these CDK mutations in both CDK9 and CDK7 seem to cause a lower cancer burden. So they slow the growth rate of these cancer cells. And this is also, we can see this if we look at whole body imaging of these mice, that there's a lower cancer burden in our CDK7, or mice that were injected with these CDK7 mutant cells. We can also see this manifest on actually survival curves where CDK7, or mice injected with CDK7 mutant cells live for longer because their cancer grows more slowly. But actually, when we normalize for these basal differences in the growth rate of these different cell lines, we actually do see that there is a resistance phenotype. So it does seem that these mutations are causing resistance in vivo, which is that they're also reducing leukemia cell fitness, which is quite interesting. So next, we moved on to subpool 2, where we tested CDK12 and CDK13. I sort of spoiled some of these results earlier. But in short, we identified dozens of novel resistance mutations in each of these proteins against 3 different inhibitors or PROTACs or molecular glues that we tested. What was quite interesting is that it seems that resistance variants in either CDK12 or CDK13 can actually cause resistance to these therapies. And the reason that was interesting to us is that CDK12 and CDK13 are paralogs. So these are very structurally similar proteins that seem to actually perform redundant functions in cells. So what we think is happening here is that if you get a mutation in either CDK12 or CDK13 that renders an inhibitor no longer effective, so no longer able to, for example, bind to that protein, that seems to be sufficient to provide resistance to these cells. Meanwhile, it's very likely that the other CDK is still likely being inhibited. So what was also interesting is we were able to map these mutations onto the structure of CDK12 and CDK13. And in particular, we identified a molecular glue specific resistance variant. So molecular glues actually mediate a protein-protein interaction, in this case between CDK12 and DDB1. And what was quite interesting is if we look at this G822 amino acid, which is highlighted in yellow, this mutation actually, or this amino acid is actually at the protein-protein interface. So it's not located in the ATP binding site. So it's likely not affecting the ability of this drug to bind to CDK12. What it's likely doing is causing resistance by getting rid of this protein-protein interaction. So we went ahead and actually tested this. So we tested a host of different CDK12 and CDK13 variants. And what we found is actually these molecular glue specific resistance variants, which disrupt this protein-protein interaction, only provide resistance to this molecular glue, but not the other CDK12 targeting therapies, which is quite exciting to us because it tells us that this approach is powerful enough to really distinguish between the mechanisms of actions of even drugs that are targeting the same proteins. So we also tested subpool 3. This is actually the most clinically relevant subpool because These are CDK4/6 targeting therapies as well as CDK2 targeting therapies that are actually in the clinic or in clinical trials. So not to belabor this point, but we once again found many resistance variants, shocker. We also validated these. So we showed if we tested these things individually, they do seem to be selected for when we expose these cells to different drugs. And this was actually quite exciting because this method of resistance to these therapies hasn't really been much appreciated. There are some mentions of it in the literature, but really the main focus is on other methods of resistance. But really up until this point, I've shown you many lists of resistance variants, and we wanted to go beyond just sort of delineating these lists. We wanted to ask, can we use these data sets to identify sites of resistance that are shared across the CDK family? So can we actually identify patterns? So back to this structure of CDK2 that I showed you. So a substrate peptide fits in there. And what's nice about the kinases is actually their ATP binding site is actually very, very highly conserved among almost all the 500 human kinases. And so there's this universal indexing system called CLIFS that allows us to directly compare the ATP binding sites of all 500 of these human kinases. And you can see why this works when we overlay the structures of the different CDKs on top of each other. These are very structurally similar molecules. And so this allows us to directly compare the resistance patterns that we're seeing among different kinases. So first, we looked at other kinases, so not kinases that aren't in the CDK family that have been drugged with different cancer drugs. And so most of these mutations that we observe in actual patients seem to occur in these ATP binding sites, which makes sense. They're disrupting the binding activity of these drugs. And we can actually map these resistance variants that we see in other kinases onto this universal indexing system. We see that there's some clear hotspots, namely mutations near the gatekeeper, hinge, and linker region seem to commonly cause resistance to these therapies. We can then overlay or map our CDK resistance variants that we saw with all of our CDK drug pairs onto this indexing system. And once again, we see that we're seeing variants in the hinge and linker region that strongly cause resistance to therapy, but we also see some other hotspots that seem to commonly cause resistance across many different CDK drug pairs. And in addition, we can move beyond just the ATP binding site, so we can look at resistance along this, or patterns of resistance along the entire structure of these CDKs by performing a multiple sequence alignment and mapping the resistance variants onto the sequence alignment. And so if we look at the actual traces of resistance variants in this alignment for each of these CDKs, you'll notice that there are differences, but there are some striking similarities where mutations seem to commonly confer resistance within these CDKs. And we can map these onto the actual structure of one of the CDKs, and essentially what we find is that there's 4 sort of prototypical sites within these CDKs that commonly are mutated to cause resistance to therapy, and those I've labeled in red. But if we look beyond the ATP binding site, we can actually identify many other resistance-causing variants, which may suggest some sort of allosteric mechanisms of resistance, which is quite exciting. And so the last thing I'm going to show you today, and I think possibly the most exciting, is that we found that the experimental behavior of CDK6 mutations predicts clinical response to therapy. So this is in collaboration with some colleagues at Sloan Kettering, where essentially we have 2 breast cancer patients here. And each of them have different CDK6 mutations, and they were each treated with a CDK4/6 inhibitor. And the patient at the top did not respond well to this drug, so they had a relapse after around 7.5 months, whereas the patient at the bottom has experienced an ongoing progression-free survival of over 7 years. So they've essentially been cured of their cancer. And what we found is that when we looked at the behavior of these mutations that we see in each of these patients, the patient that did not respond well to therapy, this CDK6 mutation actually seemed to cause resistance when we tested it experimentally, whereas this patient that responded well to therapy, they actually seem to have a sensitizing mutation. So this is quite exciting because it tells us that our findings do seem to actually be translating to how these inhibitors are behaving in the clinic, which is quite exciting. So in conclusion, we identified and validated dozens of known and novel CDK resistance variants. We validated these things in vivo. We showed modality-specific resistance variants. We also identified and generated high-resolution residue essentiality maps. That is, we quantified the behavior of these CDKs in the absence of drug, but I didn't have the time to actually go through all of that. We also performed an iterative mutagenesis approach to model the sequential acquisition of mutations that we see in patients. And we leveraged the evolutionary relationships between these CDKs to define shared sites of resistance to therapy. And finally, we show the clinical relevance of our work. So with that brings me to perhaps the most important part of the talk, which is the acknowledgements section. So to start, I really just want to acknowledge all of my coworkers, all my lab mates that really make coming to work every day fun. PhDs, it's not always easy, but it's great to come to work every day and know that I'm going to laugh at work and have a good time. So thank you guys for all of your really important contributions to these projects. I really appreciate it. I'd also like to thank the members of my thesis committee, so Michael Heeman, Jonathan Weissman, Brian Lau, as well as all my collaborators, both here at MIT and beyond, namely at Sloan Kettering. I'd also like to thank our lab funding sources who make all of this work possible. Some of these experiments, are quite expensive, so we need that funding. And I would also like to thank the core facilities at the KI that also make this work possible. I'd also like to take the time to thank my scientific mentors, so Bill Jacobs at Albert Einstein College of Medicine, Scott Lowe and Cal Sanav at Sloan Kettering, and Nancy Kleckner and her whole team at Harvard. And so Nancy's actually in the audience today, and I really want to thank Nancy in particular. She had a really strong impact on me. She really showed me sort of the level of diligence and attention to detail that's required to do good science. So thank you, Nancy. Appreciate it. So I'd also, Francisco gets his own slide. He tried to embarrass me at the start. Maybe, I don't know how embarrassing these pictures actually are. But, there's many things to say about Francisco, but really he's just been an amazing mentor throughout this entire PhD. I couldn't have imagined doing my PhD in a different lab. And, the one thing is that he really just cares about the success of each and every member of the lab. And it's really just inspiring to see how hard he's worked, and I've learned so much from him. The other thing about Francisco is that he's always overflowing with ideas. So it's not uncommon for me to come out of a meeting with Francisco with enough ideas for about 5 or 6 PhDs. So I really have to sort of pick and choose which ideas I actually follow up on. But I really want to thank you, Francisco, for all your help and for teaching me so much. So thank you. I'd also like to thank my family, many of whom are in the audience today. So my mom, my dad, my brother Owen, as well as my whole extended family, many of whom made the trip up from New York today. I don't think they necessarily knew exactly what I was doing during my PhD. Hopefully they have a better idea after today, but they've really just been incredible cheerleaders throughout the entire process. So thank you all, and I love you guys. I'd also like to thank my friends, many of whom are also in the audience. I would say that I wouldn't have been able to do it without you guys, but I think we both know that's probably a lie. But it probably would have been a lot less fun. So thank you guys for all the memories, and you know what? I love you too. So, and last but certainly not least, I'd like to thank my amazing, beautiful, kind, smart, intelligent, amazing, did I already say amazing? Girlfriend Ibby, who's really been incredibly supportive throughout this entire process. This is actually a very full circle moment because Ibby and I met as organic chemistry lab partners in Brian Lau's class, and Brian is actually the external member of my thesis committee here today. So thank you as well, Brian, for setting us up. Really appreciate that. But really, I wouldn't have been able to do this PhD without Ibby's support, and so thank you, and I love you. And yeah, with that, thank you for your attention, and happy to take any questions.
We have time for some questions. Before we really
Yeah, Yadira?
So I have a question about this statement that you made, that these mutations that you're finding in the CDKs enriched in the pockets outside of the kinase domain are potentially allosterically modulating the activity. So I'm sort of kind of fighting this idea that maybe the genomic landscape that encodes for that region is actually easier to mutate, and it's not just through biochemical allosteric modulation. I guess the question is, what is the evidence that it is allosteric modulation and not just the genetic context is easier to mutate in a way?
Yeah, I mean, so I think that would probably take more in-depth biochemical characterization to actually answer that question. I mean, we're sort of stopping at the genetic level here. One particular finding that I think was quite exciting is that actually one of those variants that I highlighted is located in an intrinsically disordered region. That seems to actually sometimes, there are some structures that show that it folds back and sort of interacts and modulates the structure of the ATP binding site. So I think that's probably one mechanism, is that there might be some sort of hidden interactions that we can maybe identify with this method. But yeah, I mean, I think to really go more in depth and understand some of those mechanisms would probably acquire additional approaches. I do think, though, that we don't really see a bias in terms of the amino acids that we're mutating. We're essentially, anywhere that there's a protospacer, we're mutating, and it doesn't seem like there's sort of GC poor regions here where we don't have protospacers. So, yeah, I mean, I think it's a good question. I think it's sort of an interesting place to follow up, but it's not something that we really got to the bottom of. Yeah, Yusuf.
So very nice work, beautiful. So the CDK7/9 mutants, do these lose function, enzymatic function?
Yeah, so the previous paper that identified the CDK9 variant with these dose escalation studies did show in biochemical assays that it does seem to reduce sort of the ATP binding activity of these CDKs, which could sort of explain some of the fitness effects.
So then the resistance would be due to possibly compensating mechanisms in, for example, other CDKs or other pathways.
Yeah, it's possible. One other thing that we showed that I didn't have the time to actually mention here is, so we also looked at the behavior of these CDK mutations in the absence of drugs, so the fitness of cells that have these CDK mutations in the absence of drug. And one thing that we found is that actually the resistance variants that we were identifying, it did seem that they actually didn't, in the absence of drug, they weren't some of the ones that sort of caused a strong fitness defect. So it does seem that you have to balance sort of gaining resistance to therapies, but since these are essential proteins, their action is essential in cells, you still need to have enough sort of residual kinase activity that these cells can still survive. Yeah?
I'm interested in sort of the sensitizing mutations that you found. And I was wondering, especially since they did seem like predictive of patient outcomes, if there's anything useful about knowing those sensitizing mutations for telling you something about the drug or ways to improve treatments.
Yeah, it's something that, the sensitizing mutations we don't focus as much on in these types of screens just because the statistical power for identifying those types of mutations, things that drop out, tends to be lower. But I do think that, it is worth, obviously what we showed is somewhat anecdotal evidence, but you could expect that there could be mutations that, for example, increase the affinity of a particular drug with a particular CDK. So that could be some sort of mechanism that caused resistance, or sensitivity rather. But it's not something that we've really sort of focused our attention on just because of that sort of statistical argument that it's much harder to identify really strongly sensitizing variants in these contexts. Great, so if there are no more questions.
If there are no more questions, we'll proceed to asking many more questions.
Yeah, class is dismissed. Thank you all.